Hello everyone. In this video we will be continuing with the rest of the anatomy and the physiology of the cornea. In the previous video we were dealing with the epithelium of the cornea. So the rest of the layers will be dealt in this video. We begin with the basement membrane. The basement membrane of the cornea is about 40 to 60 micrometer in thickness, and it has got two layers, a pale layer which is known as lamina lucida, and it has got a dense layer which is known as the lamina densa. Basement membrane is mainly made up of type 4 collagen and laminin. The subtype is alpha 5, that is called for A5. The type of collagen called 4, and alpha 2, it is seen in conjunctival basement membrane and amniotic membrane. So basically the collagen which is found in the basement membrane of cornea, conjunctival basement membrane and amniotic membrane is collagen 4, but the subdivision varies, that is the alpha 5, it is present in basement membrane, and alpha 2 is present in conjunctiva and amniotic membrane. So in the last video we were telling about the epithelium. So when an insult occurs to the epithelium, how do the epithelium proliferates back? So it is achieved by the balance between the epithelial migration, there is epithelial proliferation, and there is differentiation. So this is achieved by various changes which helps in the cell to cell interaction and cell to the matrix interaction. Also there will be an increase in the hyaluronic acid, and there is also a modulation in the extracellular matrix by the proteolytic enzymes. So let's see how this is being achieved. The cell to cell interaction and the cell to matrix interaction is achieved by the fibronectin integrin system. So let's see how it has been taking place. Okay, so as I have told, the epithelium defect repair is through the epithelial cell migration. There is cellular proliferation and there is differentiation. These things are achieved by the changes in cell to cell and cell to extracellular matrix interaction, which is achieved by the fibronectin integrin system. There is an increase in the hyaluronan, and there is increased proteolytic enzymes which modulates the extracellular matrix. Now if you see, this is the fibronectin. So fibronectin is a high molecular weight glycoprotein of the extracellular matrix that binds to integrin. This is the diagrammatic representation of an integrin, and integrin, as you can see, it is a membrane spanning receptor which attach the cell cytoskeleton to the extracellular matrix. So the fibronectin, it usually appears on the exposed site of the insult, and there is a fibronectin matrix formation which helps the cell to cell attachment with the integrin. So you can see there is interaction between the subunits of the integrin and the fibronectin. So the binding of the integrins to the fibronectin, it is mediated by something called as the RGD sequence. In the alpha subunit of the integrin, various sites like alpha B1, alpha B1, alpha 2, B3, etc. have been discovered where the fibronectin binds via the RGD sequence. So, RGD sequence means arginine, glycine and aspartate. So, where this sequence is there, the interaction occurs. So, it is mediated by the RGD sequence in the fibronectin. Now, there are various integrin subunits in the human cornea like alpha 2, alpha 3, alpha 5, alpha 6. There are numerous integrin subunits in the human cornea, and once the epithelial migration has occurred, the hemidesmosomes reappear, and the hemidesmosomes which helps in the cell to basement membrane attachment, and the component of this is for hemidesmosomes. For hemidesmosomes, it is the integrin alpha 6 beta 4 heterodimer, which is not related to this fibronectin mediated cellular interaction. It is not mediated by fibronectin. Also, I have told there is increase in hyaluronic acid. So, trials are going on in the eye drops which contain the hyaluronic acid, which can promote wound healing, and there is increase in proteolytic enzymes. Other than these, there are also expression of cytokines and growth factors, which includes growth factors like EGF, TGF beta, FGF and PDGF. There is also synthesis of interleukins like interleukin 1 and interleukin 6. So, coming to EGF, EGF is basically synthesized in the eye in the lacrimal glands. It stimulates cell proliferation and cellular adhesion to the fibronectin matrix. TGF beta 1, it is synthesized by corneal epithelial cells. It helps in cell migration. It modulates the effect of EGF on the epithelium for the attachment of the cells to the fibronectin. FGF, it binds with heparin sulfate, which is present in the basement membrane, and it also helps in angiogenesis. The receptors for FGF is present in the stroma. Coming to PDGF, PDGF is present in the tear fluid. Coming to interleukin 1, it helps in the healing of epithelial wound. Interleukin 6, it stimulates the expression of integrin alpha 1 beta 1, and it increases cell proliferation. So, it regulates the epithelial migration. and it modulates the fibronectin integrant system. Coming to the bowmans layer, the function of the bowmans layer is that it acts as a base for the epithelium uniformity, so that it actually helps in the refraction. And as you all know, it is acellular. It is a membrane-like structure with approximately 12 micrometer thick. There is random arrangement of collagen fibers and proteoglycans. The collagen is mainly type 1 and type 5 collagen. So the anterior part, which faces the basement membrane, is the smooth, and the posterior part appears to be continuous with the stroma, and the bowmans layer do not regenerate. Next, coming to the corneal stroma, so it contributes to more than 90% of the thickness of the cornea. The stroma is also called as a substantia propria. It is around 0.5 millimeter of thickness. As you all know, it is composed mainly of collagen fibers, which are regularly arranged, and cells known as keratocytes, and there is also a matrix which is composed of proteoglycans. So the keratocytes are basically derived from fibroblast, and when there is an insult or an injury, the cells migrate from around the blood vessels to the site of the injury, and these type of cells are called as wandering cells. Also, some of the keratocytes, they get differentiated into myofibroblasts, which also helps in wound healing by laying down of collagen, extracellular matrix, then generating cytokines and MMPs. So let us see in detail about stroma. So as I've already explained, stroma is also known as substantia propria, which contributes to more than 90% of thickness of the cornea, which constitutes around 0.5 millimeter of thickness. There is a uniform arrangement of collagen fibers, cells and proteoglycans. Coming to the cells of the cornea, they are mainly keratocytes, which are basically fibroblasts, and it contributes to 3% volume of the stroma, and the cell turnover is 2 to 3 years. They are spindle shaped cells. There is tip to tip connection of the cell with gap junctions. They have prominent actin filaments containing intracellular cytoskeleton. The intracellular cytoskeleton has got actin filaments, and in any case of insult, they are converted into myofibroblast, which expresses alpha smooth muscle actin and which helps in wound contraction. Now, I've already told about the wandering cells which migrate to the site of the injury, and the myofibroblasts, they lay down extracellular matrix, collagen degrading enzymes, MMPs and cytokines. Talking about the MMPs, the MMP2, which is also called as gelatinase A, is the one which is present in the normal cornea, and the others like MMP1 or collagenase 1, MMP9 or gelatinase B, and other MMPs like MMP 7, 8, 9, 11, etc. are produced by the cornea or the epithelial cells at the time of any injury. So the stromal cells, the stromal tissue, thus it gets the repair and the contraction of the wound by all these mechanisms. Now coming to the collagen. So more than 70% of the dry weight of the cornea is due to the collagens. It is mainly type 1 collagen, but there is also type 3, type 5, 6, 12, 14, etc. in the corneal stroma. So because of the homogeneous arrangement of the collagen fibrils, the light rays are allowed to pass through it. So it is basically produced by keratocytes. The keratocytes produce something known as the procollagen. The procollagen, it loses the propeptide, and mature collagen is formed. These mature collagens assemble to form collagen fibrils, which are around 10 to 300 nanometers size, and these fibrils get together to form fibers. So the diameter of a fibril is 22.5 to 35 nanometer average, and the distance between the fibrils is around 41.4 plus or minus 0.5 nanometer, and each fibril has cross striations, and around 300 collagen lamellae are formed from the limbus to limbus. And coming to the lamellae, there is an anterior lamellae and a posterior lamellae. Anterior lamellae is interwoven. Posterior and mid lamellae, they are parallel, and it is because of this interwoven lamellae which gives the anterior stromal curvature. The turnover of the collagen is 2 to 3 years. Now coming to the proteoglycans. So proteoglycans constitute a major component in the extracellular matrix. So this proteoglycans has the ability to retain water. So in cases of diseases like mucopolysaccharidosis and all, there is more extracellular matrix, there are more proteoglycan deposition in the cornea, which can lead to defects in the corneal, which can lead to corneal edema, or there is some loss of translucency of the cornea. So the proteoglycans, as the name suggests, it is mainly composed of a core protein and a glycosaminoglycan, and as all of you know, the most abundant proteoglycan is keratin sulfate. In a normal cornea, the proteoglycans are synthesized by keratocytes, which are in the stroma, but in the early phase of injury, the epithelial cells can also synthesize proteoglycans. So when we see about hyaluronan, which is an exception, it does not contain a sulfate group, and it is not found covalently attached to a protein. So among the other proteoglycans, hyaluronan is an exception. Now there are various core protein. The core protein is small leucine rich proteoglycans. So there are a core which contains the leucine amino acid, and the coproteins are lumican, keratocan, mimican, and dicorin. So we will be discussing about the proteoglycans, the function of the core proteins, and the different types of proteoglycans in brief. So as I have told, about around 65% of the proteoglycan in the extracellular matrix of the corneal stroma is keratin sulfate, and the proteoglycans, they also modulate collagen fibrillogenesis, Now coming to the core protein, which are small leucine rich proteoglycans. Core protein. So basically we can divide them into keratan sulfates, chondroitin sulfate or dermatan sulfate core protein. So in keratan sulfate, the core proteins are lumican, keratocan, mimican or osteoglycin. In chondroitin sulfate and dermatan sulfate, it is decorin and biglycan. And the importance of keratocan is that the mutation of keratocan leads to cornea plana. There is loss of curvature of the cornea. So these are the core proteins and these are the proteoglycans. Now lumican, it interacts with the corneal epithelial cells and it also helps in organization of collagen fibers. Decorin, it helps in wound healing. Now this is a table which is there in Kretschmer. So this table basically it shows you the glycosaminoglycans, their size in kilodaltons and the disaccharide which is contained in it. So as you all can see, basically the first part is N-acetyl galactosamine and the second part differs. In heparin sulfate, it is glucuronic acid. In chondroitin sulfate also it is glucuronic acid. In heparin also it is glucuronic acid. In hyaluronan it is glucuronic acid, but in dermatan sulfate it is iduronate acid, and in keratan sulfate it is galactose. Because of these glycosaminoglycans, the stroma is able to hold water and the normal stromal swelling pressure is 60 mm of mercury. Other than glycosaminoglycans or proteoglycans, there are also other constituents in the stroma like enzymes for glycolysis and Krebs cycle. There are also soluble proteins which contributes 5% of the stroma, which includes albumin, immunoglobulins, and these immunoglobulins are derived from the serum. So as I've already told, when there is an injury, there is transdifferentiation of keratinocytes in the myofibroblast. There is also upregulation of proteolytic enzymes, matrix metalloproteases. There is laying down of collagen, and the hemostasis of the corneal stroma is achieved by a balance between synthesis and the degradation of extracellular matrix. And there are also synthesis of various cytokines and growth factors which trigger the activation of various signal transduction pathways. So in case of a corneal ulcer, there are various mechanisms by which the defect can occur. One is by direct degradation by bacterial collagenases. Second is by activation of matrix metalloproteinases, example is Pseudomonas, which has both of these actions. That is, it can have a direct action on the cell and it can also activate MMPs. And of course, the inflammatory cells produce proteases which degrades the cell. The keratocytes, as all of you know, they differentiate into myofibroblast, and TGF beta has got a key role to play in the healing of the stroma. This is expressed by the epithelial cells stromal cells and the other inflammatory cells. This promotes the transdifferentiation to myofibroblast, which helps in wound repair and also in scarring. Coming to the descements membrane. So descements membrane, it basically represents the basement membrane of the corneal endothelium. At birth it is 3 micrometer, and in an adult the value ranges from 8 to 10 micrometer. It has got a thin non-banded layer near the stroma, an anterior banded zone and a posterior amorphous zone which constitute more than 4 micrometer, that is around 2/3 of the thickness. Now it is mainly composed of type 4 and type 8 collagens, laminin and fibronectin. This type 8 collagen is produced by the endothelial cells. There are no glycosaminoglycans in the descements membrane. Now any rupture in the descements membrane can lead to aqueous into the stroma, which can lead to the corneal edema, and also there are excrescences in the descements membrane. One is called as Hassel-Henle bodies, another is called as guttata. Hassel-Henle bodies are peripheral excrescences and guttata are central, and they increase in number as the age advances. So the posterior surface, there are wart-like excrescences in the periphery called as Hassel-Henle bodies, and in the center there are excrescences known as guttatae. Now coming to the predescements layer or the Duas layer, it is basically 15 millimeter thick. It is acellular and it is mainly composed of type 1 collagen. The other collagens are type 4, 6 and 5. There are no keratocytes. There are proteoglycans present, and unlike the descements, they do not extend to the periphery. Next, coming on to the corneal endothelium. So as you know, endothelium consists of they consist of cells which are polygonal or mostly hexagonal type of cells, and endothelium is very important because it has got various type of pump mechanisms which prevents overhydration of the corneal stroma. Also, you know that the cells, which are around 3500 cells per millimeter square in an adult, it keeps on decreasing as age advances. So a cell count less than 500 cells per millimeter square is a grave condition which can lead to the swelling up of cornea and corneal decompensation. So there are two things called as polymegathism and pleomorphism. As there is cellular loss in the cornea, the cells tries to compensate for them, the residual cells. So polymegathism means there is increase in the size of the cell, that's variation in the size of the cell. Pleomorphism means the variation into the variation of the cell shape. Okay, so let's see more about endothelium. So as I've told, endothelium is a single layer mosaic pattern cells. They are present at a rate of 6000 cells per millimeter square at birth. Each cell is around 5 micrometer thick, 20 micrometer wide. They are polygonal or hexagonal, and in a young adult, the cellular count, the average is 3500 cells per millimeter square. The cell count falls by 25% in the first year and 26% the first year and 26% in the next 11 years. And in the old age, because of the dropout of the cell can increase in size, and it can reach up to 400 micrometer in size because the cell tried to compensate for the loss. Now, as I've told, polymegathism, it means there is increased variability of the cellular area, and pleomorphism means there is a deviation from shape. Now, the endothelial cells is highly active cells. So, of course, they have got a large nuclei, they have abundant organelles, they have gap junctions for passage of small molecules, zona occludens, macular occludens, etc. And they basically produces a barrier function. So, if there is a loss of more than 75% of the cell, or there is less than 500 cells per millimeter square, corneal decompensation occurs, and these are attached to the descements membrane by hemidesmosomes, and that laterally they have interdigitations. Now, the endothelium has got various ion transport mechanisms like sodium potassium dependent ATPase. There is a sodium hydrogen exchanger. There is a bicarbonate system which is being led by the enzyme carbonic anhydrase. So, sodium potassium ATPase is activated by steroids and insulin. Now, the endothelial cells, they do not proliferate. So, once an injury has occurred, the lost cells cannot regenerate back. So, I am ending this video here. We are left with a few topics like nerve supply of the cornea, blood supply of the cornea, and the anatomy of the limbus, and the mechanisms by which the endothelial cells maintain the barrier function. So, all those topics will be discussed in the upcoming video. I hope you enjoy this video. Thank you.